Okay, let's talk about nucleic acids. So we're going way back in the book to chapter 3. We're looking at chapter 3.5, which is specifically about nucleic acids. And after this chapter, you should be able to describe the structure and the two types of nucleic acids, DNA and RNA. You should also be able to explain DNA and RNA's roles and structures and the difference between the two of them. Okay, let's jump in. Nucleic acids are the most important macromolecules for the continuous for life, essentially. And this is crazy. Like, let's just wrap our mind around for a second that all of life, every cell, all of it, from wheat to mice to humans to everything, everything is made up of these four nucleotides, right? Cytosine, thymine, adenine, guanine, 4 letters in the alphabet of life. 4 letters make up the complexity of life. It is mind blowing, at least to me. I'm like, what? We have proved right in the last chapter that it is really DNA. It is the 4 nucleotides that make up this life, and it is just crazy to think about that. Essentially, nucleic acids carry the cell's genetic blueprint and carry instructions for the entirety of life, and it is just crazy. So they are made up of 3 parts. We talked about this in some of the earlier chapters. You have this phosphate group here on the end, a central phosphorus atom bonded to 4 oxygen atoms. There are 4 oxygen atoms here. They're just not shown. A pentose sugar, a 5-pentose, a little, taking it back to organic chemistry, 5-pentose sugar. Now, deoxy, D, 2 oxygens, has 2 oxygens. Ribose just has 1 oxygen. So, deoxy, 2, just regular RNA, ribose, just 1 oxygen. And that is on this sugar molecule where the oxygen is. Now, the nitrogen bases are going to be your adenine, guanine, cytosine, or thymine in DNA, and that's going to be this base connected here. Each nucleotide in DNA contains one of the 4, right? Either adenine, guanine, cytosine, and thymine. And those are the purines or the pyrimidines. So we classify adenine and guanine as a purine. That's just the name that it has. That's little organic chemistry classification. The purine's primary structure is a 2-carbon nitrogen ring. So let's look at this for a second, right? There's 2 rings, 2 carbon rings. Those are purines. Pyrimidines just have 1 single carbon nitrogen ring. So that's the difference between the purines and the pyrimidines. Each of these basic carbon nitrogen rings has different functional groups attached to it. Okay, again, in molecular biology, the shorthand for this is ATCG, right? Adenine goes with the thymine, cytosine goes with the guanine. And so DNA contains A, T, C, and G, and RNA will contain this uracil here instead of the cytosine. And there is the bigger picture of that. The 2 main types, again, are deoxy, the 2 oxygens, and just the ribonucleic. DNA in eukaryotic cells, but not in prokaryotes. Remember, in prokaryotes, it just twists and winds around itself. DNA forms a complex with histones, right? Our DNA is going to wind around these histones to form chromatin, and that chromatin is going to form chromosomes. See what they did there? Chromatin, chromosomes. And the genes contain the information to make protein products. DNA controls all of these cellular activities and can turn genes on and off. So the other type of nucleic acid, RNA, is mostly involved in protein synthesis. And there's going to be 3 types of RNA that we're going to mainly talk about. DNA never leaves the nucleus. DNA stays in the nucleus. So DNA is going to need a messenger to get from the nucleus to where these proteins are going to be made. So think of the DNA as a library book that cannot be checked out, right? You can't check it out of the special section of the library. It cannot leave the library. So the DNA never leaves. It has to make a copy with the messenger RNA. So your DNA will unwind and transcription will take place. Transcription is DNA to RNA. Now, when you think about transcribing or transcribing something, if I said, can you please transcribe my notes? That would be in English. You would just copy it. You would copy my notes. That's transcribing. DNA to RNA is transcription. Now, if I wanted you to translate my notes, that would be in a different language. That's translation, and that's going from RNA into amino acids, ultimately into protein. So we'll come back to that again. But transcription is DNA to RNA, same basic language. Now, the pentose sugar in DNA is deoxy, 2 oxygens. See, here we have this oxygen. Both of them have this group here, but your deoxy has 2, your ribose has the other oxygen. The difference between the sugars is the presence of the hydroxyl group on the ribose's second carbon and hydrogen on the deoxy's second carbon. Okay, so deoxyribose, this is the oxygen that we are talking about. The carbon atoms of the molecule are numbered 1, 2, 3, 4, 5. We already did this. Remember? 1, 2, 3, 4, 5. That's how we're numbering the carbons. Okay? 1, there's a carbon here. 2, 3, 4, 5. Okay? This is your 6th carbon up here. The phosphate residue attaches to the hydroxyl group of a 5' carbon. That's our carbon number 5, right? 1, 2, 3, 4, 5. Okay, so the number 3 carbon of the previous group attaches with this phosphodiester linkage to the 5' carbon. Okay, do you see? We're numbering the carbons. 1, 2, 3. So the 3' or the number 3 carbon is attached to this 5' that's kind of hanging out here, right? It's not part of the the cyclo. It's not part of the chain. Okay, so you've got the 5' connected to the 3'. A simple dehydration reaction. Do not panic when I say the words dehydration reaction. It will be okay. When you dehydrate something, you just remove some water, like when you're dehydrated, right? So we're just removing some water here with a dehydration reaction. So a simple dehydration reaction, like other linkages, connects these monomers together and connects our phosphodiester linkages to create our DNA. So a phosphonucleotide may have thousands of these phosphodiester bonds, right? This is putting our bases together from 3' to 5'. Right, here's our 5' connected to the 3', and then we're doing that with the dehydration reaction where we're going to lose some water. DNA has a double helix structure. We already knew that. The sugar and phosphates lie on the outside. So here's our sugar and our phosphates. And what are these guys? These are our nitrogen bases. These are our A, T, C, G. And this makes like a winded staircase, just like this. Every base pair in the double helix is separated by the next base pair by 0.35 nanometers. Okay, we can measure these things, right? So here's this in between this one and this one is 0.34 nanometers. The whole turn is 3.4 nanometers. Isn't that crazy? That's just like so beautiful when the math works out. It just makes my heart happy when the math, looks neat. The helix is 2 strands run in opposite directions, right? This one goes here, this one goes here, right? And they come together like this. One will face that 3' carbon and the other will face the 5' carbon. We'll look at that in just a minute. Scientists call this anti-parallel. Anti-parallel, and it's the orientation, and it's important for DNA replication. Only certain types of base pairing are allowed. You have to have this pairing with a pyrimidine or it wouldn't work, right? You have to have thymine has to go with adenine. And this is what this linkage is right here, okay? This is what we're going to pull apart when we go to replicate the DNA. So A has to pair with T, C has to pair with G, and this is called base complementary, right? Chargaff's rule, base complementary, whatever you want to call it to help you remember. A goes with T, C goes with G. If the sequence of one strand is AATTGGCC, like I'm going to give you these and you're going to have to give me what the other side of the DNA would be, then the complementary strand, cannot say the word strand, right, is going to be TTAACCGG, right? And this will look a little bit different if it's RNA, but we're not there yet. We're still talking about DNA. During replication, each strand copies itself, resulting in a daughter DNA with a double helix containing one new strand and one old strand. Okay, so the ribonucleic acid, or RNA. So we know how the DNA gets replicated. Now we need to make RNA. So the ribonucleic acid, or RNA, is mainly involved in getting the DNA from the library or the nucleus out and then making proteins from it. Okay? So RNA is usually single-stranded and is composed of ribonucleotides that are linked by a phosphodiester bond. A ribonucleotide in the RNA chain contains ribose, the pentose sugar, we already talked about that, one of the four nitrogenous bases, A, U, G, C, no thymine, just uracil, and the phosphate group. There are four main types of RNA. Messenger RNA, which we're going to talk about first, and that's just the copy, the photocopy, right? If you go into the library and you make a copy, that's what I would think of your RNA as. Like, okay, I have a copy. I know what it should say. RNA, rRNA, ribosomal RNA, is what's going to make this little hamburger-looking thing that's going to actually put everything in order so we can translate it. The translated mRNA will come in so we can transcribe it. And then we have tRNA, which are these little things here that actually connect the mRNA molecule to the amino acid to make that chain of amino acids. That is my pug snoring, just in case you would like to know. The first mRNA carries this message and then it grows a chain. Let's look at it. The RNA base sequence is complementary to the DNA's coding, right? A goes with U, G goes with C. However, in RNA, there's no T, like I just said. It's only U. So let's do this again, just like we did with DNA. If your DNA strand is AATTGCGC, your RNA will be UUAAGCCG. Okay, what if DNA was TAGGCCAAAGCGT? Okay, so pause it, see if you can do that one. It's pretty easy. Okay. The mRNA is read in sets of 3 bases. So once we get here and we've transcribed it from DNA to RNA, we're going to have this string of mRNA. Now we need to translate it into a different language, which will be the language of amino acids. You are going to look, each one of these 3 codons codes for an amino acid. Okay, so there's a chart for that. I'm not going to make you memorize it. And you will have a chart and you will be able to do this. The mRNA is read in sets of 3, known as codons, and each codon codes for a single amino acid. In this way, the mRNA is read and made into a protein product. This squiggly green madness is the rRNA. That's what actually puts everything together so it can make this long string of amino acids. It holds everything together. It's the little house that brings everyone together. The rRNA ensures the proper alignment of the mRNA and the ribosome. The ribosome's rRNA also has an enzymatic activity, right? Peptidyl transferase, which catalyzes the peptide bond, which forms between each one of the amino acids. Okay, transfer RNA, again, here's just another way that you may have seen it looking. So you're going to have, this is your tRNA, okay? This is your ribosomal subunit. This is your mRNA, okay? mRNA, rRNA, the purple, and then here's our little tRNAs, because they do look like little t's, okay? And they're going to have our codons, and in the end of that, you're going to see this growing protein, okay? They're usually 70 to 90 nucleotides, these 70 to 90 nucleotides for these tRNAs. And it is causing the base pairing between the tRNA, that's this, and the mRNA that grows this protein chain. MicroRNAs are also these tiny RNA molecules it's essentially regulating gene expression. How and when we have gene expression is become incredibly important, especially in the world of epigenetics. And it interferes with the expression. It can actually prevent the expression of certain mRNA molecules. And here is a chart breaking it all down. Here is another view of our mRNA, tRNA, and this is the growing peptide chain. The DNA dictates this structure in transcription, right? Going from DNA to RNA, same language, and this all happens in the nucleus. Then we leave the nucleus with the mRNA, and that grows into a protein. This is called the central dogma. It really is just DNA to RNA, RNA to proteins. That's what we mean when we say central dogma. Now, can you describe nucleic acid structure and define the two types of nucleic acids? And can you define DNA and RNA's role in transcription and translation? Okay, sorry that video was a little long.